This is Dr. Carotenuto. I'm presenting this poster to the AMMF Congress of 2021. I will briefly present our data supporting our project. As you can see from the title, we perform an integrative analysis at genomic and transcriptomic level of several samples of bile duct cancer, obtaining some novel key genes and proposing a new therapeutic approach. Briefly, bile duct cancer is a malignancy thought to be derived from cholangiocytes, and it is increasing worldwide. Now accounting almost 10 to 15% of whole hepatobiliary malignancies. The mechanism underlying the cholangiocyte malignant transformation is not completely understood. And we thought that genomic profiles of cancer patients may lead us to clearly understand the carcinogenesis, to obtain a good classification of patients, and to obtain new therapeutic approach in this aggressive form of cancer. We briefly we analyzed around 1,000 of bile duct cancer samples obtaining from an Italian cohort. We performed a next-generation sequencing approach to analyze genome and transcriptome of these of tumors coming from these patients, and our bioinformatic pipeline led us to obtain some new data, especially to obtain some new target genes in this tumor. We performed a macro dissection for each tumor sample, obtaining RNA from tumor, stromal, and normal area. And we analyzed both the genomic, both the transcriptome landscape of these areas. Interesting, we obtained several differential expressed genes, but more interesting, we obtained that a lot of genes belonging to primary cilium genesis are differentially expressed, especially between tumor and stromal area. Belonging to them, several cilia-associated genes such as OFG1, CNGB1, and others. We started to study at functional level some of these genes, especially OFG1, and we demonstrated that OFG1 depletion in cholangiocytes increased the number and the length of cilia. Furthermore, the depletion by siRNA interference of OFG1 decreased the number of viable cells in cholangiocarcinoma cells. We next performed a high throughput screening strategy to obtain specifically compounds targeting ciliary-associated genes. And we definitively obtained some compounds, compounds that could be proposed as novel therapeutic approach. In particular, our hypothesis is that the ciliary formation, the ciliogenesis, could be used as tumor suppressor in cholangiocarcinoma, and other bile duct cancers. We obtained 2 compounds that can be used to enhance cellulogenesis in cholangiocarcinoma in vitro experiments, and we aim also to analyze these genes also in animal models, these compounds and their target genes in animal models, to obtain more clinical relevant data about the newly identified compounds. Thanks a lot for your attention, and see you at AMMF Congress.